Sounded. Okay. So cancer biology. So many different types of cancer, and they're all um, and
There's so many variables.
so many
So many ways
different.
that it's gonna be very challenging.
Because it's basic. It's yours. All of three things. Well, depending on if you're a blood cell or not. So the definition of cancer, at least for molecular, is when a cell and then when cells start to control what should happen, dying. Cell suicide. And it's a normal process. Undergo apoptosis, but they don't. So cancer cells, besides having increased mitosis, then this is really only true. But they don't change. They exhibit a decrease in differentiation. This is what cancers, they divide out of control, they don't kill themselves, and they don't change. Now, let's say a cell in your body starts accumulating the errors, the problems to turn into a tumor cell, right? So now that cell starts dividing out of control and it forms into a tumor, right? That's not the end of the world. Any quack surgeon can go in and take a tumor out, right? That's not what becomes mostly or usually fatal about cancer. So, yes, who's taking physiology? So,
Well, animal.
it's all right. So let's say we have some tissue here, right? We got a little blood vessel. Right, so the capillaries are pressure is constantly leaking out of the vessels into the interstitial space. Right, so the fluid that's in the interstitial plate collects into these other vessels called lymphatic vessels. Right, and these lymphatic vessels, yeah, right. The diffusion of of flat. Developing a tumor in the interstitial space, pieces of those are set off the lymphatic vessels, and then they can spread all over the body. And you imagine like this, and just that's possibly a. And now the tumor has metastasis. Oh, really dicey. So is one of the it's when yeah. So we'll just prevent. But it's good. What needs oxygen? So there are factors in the cells that induce the production of blood vessels. Talk about. And this is like dark pink and the light pink cell, the network. A local induces tumor themselves. There's a lot, sir, but more likely out of this course. Affecting is a figure. Also going to talk about it will induce tumor. Okay, so oh, and it happens on both chromosomes. Now you have a tumor. Right? The chances of that happening are pretty slim. That's why glioma is very rare. Lineage of it. Where you inherit one of the mutations, now you only have to go through one round of somatic mutation. And you have this sort of cancer. So the And get this. All right. Total idea. Different cell lineages, right? So you got your cell progressive per cell, right? So complex. So you have genes, have what we call tumor suppressor genes. So for an oncogene to form, it's basically a proto-oncogene. This is a normal gene that we all have, that we all need, that plays an important role typically in an oncogene. And what's happening is that gene is like tumor suppressor, like cell division, or they promote things like apoptosis or differentiation. So increase apoptosis. Right? Don't get confused with apoptosis. In cancer studies, apoptosis is good because you're killing the cell. Increase differentiation. Okay. Cell division. And potentially could decrease apoptosis or decrease differentiation. All right. Also, tumor suppressor because inhibition, right? Nothing that's slowing down the cycle. Out is think. Here's some need some memory of these genes and the type of cancer genes. You know, RAS mutated very well. A person means that they have one of the alleles here that doesn't function like it should, right? The recessive allele. It doesn't function good to decrease cell division, increase apoptosis, and increase differentiation. So if one of these gets knocked out through a translocation or a deletion, now you have just that crappy copy of the allele, and that's enough right there to induce cancer. Those are your two hits for the tumor suppressor pathway. Common tumor suppressor. Big and lung. Complex these things get, right? So, but these are all different signaling pathways. Like there's RAS right there, and you know they respond to different post-translational modifications. Like, do they get phosphorylated or glycosylated? And that response is in response to usually ligand binding. So something binds to a receptor on the cell, phosphorylates and activates the protein, and then it ends in a change in transcription of genes. Okay, so why are oncogenes usually dominant, whereas tumor suppressor genes are recessive? Correct. It's a single one, right? Main thing of it And also the oncogene, right? Right, they're driving. Those three. If they're active. That's obviously cancer biology because the cell cycle is what regulates the division of the cells. So what are the stages of the cell? G1, S, G2, and M, where M is active mitosis. So here's the life cycle of the cell. And it's where the cell is kind of getting ready. The S stands for one, and then at some point, it makes a decision to divide, right? Mitosis. Is a checkpoint called the G1S check. Makes that decision. There's a few more checkpoints, but that's the checkpoint of all of them. There's a checkpoint here, the SG2, checkpoint here, the G2M, and there's a checkpoint here, anaphase checkpoint. Okay, so this, all the DNA is getting copied. All the DNA gets copied, right? Because each daughter cell needs to get a copy of the. So what we're going to look at is a pathway that's involved in this checkpoint today. So if we checkpoint, then the cell's always going to be going like this. This checkpoint here involving the assembly of the two molecules. Proteins are called cyclins. And cyclin dependent kinases. What do kinases do? Kinases are learn this. This forward things. That'll be on the end cap. So these two proteins here, cyclins and cyclin, modulate these checkpoints. There's the cell cycle, you'll see different and in kinases. You'll also see where they go up. Regulating the G1S checkpoint. Right. RB will go and bind to DNA and drive transcription of the genes that are needed for. Right. So when E2 factor binds to And if it, that would be, right? When RB, and do that promoter region, and you're always bringing this in. Very classical. I've taken out a, we'll cover the rest of it next week. That chapter, so the rest of the stuff. Yes, ma'am.
Can you go over again if there is mutations?
Suppressor. So that drives transcription of this, turn on the cell cycle. RB is mute. You have cancer. You okay?
Example, what I would-
Yeah. So there is a protein on mitochondria called BAX. This pathway phosphorylates BIM. BIM goes to the nucleus, and it's a transcription factor that turns on genes that decrease apoptosis. Right? So that's an example for a pathway. So here you have oncogenes. We'll call them since I'm. The two proto-oncogenes. So and hyperactive, now we're just kind of blowing up this whole pathway. And we're getting more transcription of genes that. Okay. Hold on, let me.
On the oncogene and the tumor suppressor, does it have to be all three? Will it affect all three of those or can it only affect?
Most cancers metastasizing one.